Thank you. Could I ask the speakers to take their places for the question and answer? We've got 10 or 12 minutes for that, so if there are any questions in the room, and will there be questions online, potentially? Yeah. So, can we have in the middle first and then at the back?
Hi, thanks. I'm Corinne Whitby from the University of Essex. This is a question for Sarah. So really good talk, Sarah. I was interested in, you mentioned doing some chemical weathering experiments, and I wondered if you could maybe elaborate a little bit more on that, and maybe ask you, are you considering doing some biological degradation experiments to maybe speed that process up?
Freya, who's in the audience, who just passed you the microphone actually, she is doing that work for us, so it might be good to pick her brains a little bit more about the chemical weathering element, but I can tell you that we are doing FTIR analysis to look at the polymer and how we've got the samples underneath, so the protected stuff that's been within the pipeline versus what's been exposed to the environment. We're doing comparisons between the two to see if there's differences in the polymer, and we can look at chemical weathering. We're also going to be doing some hardness testing and some hopefully some pyrolysis as well to look at leachates and things like that as well. In terms of the biology, the samples were pressure washed as they were removed from the environment, so there's not much that we could do there, but it's something that we'd be very interested in. And actually the microbial things as well, that's really interesting to us in terms of how the plastic might be being weathered by the biological community. So not at the moment, but it's something that would be good to investigate.
Hello, Anthony Knights, University College Cork. Question for Ben, actually. I'm not a molecular ecologist, so apologies for my naivety if this is not the case. I didn't think eDNA could measure abundance. I thought it was purely presence-absence. So could you clarify that for me? That's really...
Yeah, so kind of the output of the DNA sequencing data is you have counts for how often that particular DNA sequence occurs within your sample, and then from that you can kind of calculate changes in the relative abundance, not absolute abundance. So you could alternatively use a different approach, qPCR, to get a rough idea of the total number of that gene, and then you could kind of apply the relative abundance number to that. So if I calculate 100 million 16S genes, and then I see the relative abundance is 40%, that could give me a rough estimation.
So how do you, have you validated that then? I mean, I've seen in other cases where they've done it for fish, for example, where they've actually gone out and sampled true numbers and correlated that to...
So as Nat showed, we've got the macrofauna data, and we're going to analyse the 18S data and then see if there's any parity or disparity between the two measures, basically.
Thanks.
Okay, if we can take the next question online, please.
Yes, great, thank you. We've got a question from David Santello. For Natalie, interesting approach to analysis of existing data. Have these analyses also been able to include meiofauna, and are meiofauna communities included alongside macrofauna and microbiota in the more recent studies?
For our project, no, we haven't looked at meiofauna. We could probably pick that up with the genetic approaches. But for Zelen's work, which I think is what he's maybe interested in, Zelen went through everything that was in the database, and there are some meiofauna there too. So if you have a look at his paper, for every species, he's assigned at different trophic level, and it's things from detritivores to filter feeders to predators. So yeah, some of that is in there, but meiofauna is a big gap, and I know within the UK we don't have a huge number of experts for identifying that, so it's definitely something that's missing.
Are there other? Right.
Yes, there's one here for Sarah. So what's the sort of relative contribution you think of sorry, David Patterson, University of St Andrews. What do you think the relative contribution or likely relative contribution of plastics from degradation from oil and gas is compared to the amount that we're putting in from land and from other industrial processes? Thanks.
Yeah, it's a really good question. It's something we've been trying to consider, because obviously that is very relevant. It's not something we've managed to get actual numbers on yet, so we don't know what proportions we're talking about. But I think the overall point is that regardless of what the contribution is, it's still going to be plastic that is persisting in the environment for hundreds to thousands of years, so it's adding to that standing stock, and we know that the standing stock of plastics in the environment is just increasing. So I guess the focus is on trying to think about how we can reduce it on a global scale from all the sources. So, but yeah, we will be trying to put that into context with our report. We touched on it a little bit in the first phase, but it's quite difficult to quantify it. But yeah.
Thank you. Questions in the middle here, please.
Hi, I'm Jordan from the University of Hull. I have a question for Natalie. So obviously you said that the data that is often collected by industry doesn't benefit a lot of what we're trying to get out of it, but what do you think of seasonality in terms of the data that's collected by industry compared to what you're then getting? Is it matching or?
Yeah, seasonality is a good point, and it's something we think about. And I think it depends what metrics you're measuring as well. So, for example, we measure carbon quite a lot. And while at depth, that carbon might not change on a seasonal aspect, we know it does on the surface with blooms as well. That's not really something that has been captured. We know from Zellen's work that sometimes the time that that data was collected has been included. Other times it's just a year. And so, yeah, we don't have a good handle on that. But you're right, for some things it'll be really important.
Thank you.
If we can take the next one online and then in the front there, please.
Okay, thank you. Natalie, it's another one for you, I'm afraid. From Amit Jorgensen online. She's wondering if you were also able to compare the change in biodiversity and blue carbon pre and post construction with the situation after decommissioning. And it would be interesting to know what extent biodiversity recovers after decommissioning.
Yeah, I'm all on board for any more blue carbon work for sure. The difficulty with carbon measurements is it needs to be collected for carbon. So for things like the macrofaunal, you can grab and get a chunk of sediment. For carbon, you need that sediment profile so you can look at the amount of carbon in those layers, but you also need that sedimentation, and we don't tend to have that with industry data too. So it is a bit of a gap, and for us personally, we'd love to see that change because the seabed's being changed into how it's used around these oil and gas platforms. You don't have things like trawling going in, so we think that's affecting some of the baseline dynamics, but that's a time snapshot we don't yet have, and we'd be really keen to get.
Hi, Josh Lawrence again from Heriot Watt. A question for Natalie and a bit for Ben. Something we've been struggling with a little bit in fish spans is the concept of a baseline or background levels, and sort of we're thinking about fish densities. But I was wondering how happy you are with 3.2 kilometers being far enough away to be completely outside the impact. Like there was on some of the plots you showed, like the echinoderms, it was increasing as you moved away and then maybe dropped off. And I think the same with the pH, it was dropping and then increased to the furthest distance. How can you sort of attribute that to the platform versus patchiness globally across the North Sea? And are there other data sources that you can use to try and establish a robust baseline?
Okay, I'll start and then feel free to jump in. Yeah, no, absolutely, you're right. And when we did this, we were trying to make sure we had a control site that represented the same hydrodynamic sediment type as well and would be far enough away from the platform to be outside of that footprint. One of the things we have done is we've used radiochemistry in the sediment samples, looking at different radionuclides. So we're looking at lead-210, cesium, and radium, and those are elevated and produce or discharge water from the platform. And we can actually measure how far that impact has gone around the platform. So we know for Northwest Hutton that's extended to about 200 meters. And then beyond that, we don't have the impact of the platform. So in reality, anything perhaps outside of that exclusion zone would be a good control. So that's been quite a useful and unintended aspect that we found during the project. Do you want to add anything? Yeah. Does that answer your question? I think that's the baseline thing is really difficult for sure. And of course, there's other things going on. As soon as you move out of that exclusion zone for an active site, you've got other activities happening on the seabed which are having that impact. And we're seeing some of that with the changes in biodiversity. So you're now into areas that are trawls, so the biodiversity is going to be monitored anyway. So it's almost like when Hugo talks about his work, it's almost like a halo effect. So beyond that 200 meters and before you get outside that 500 meters, you've almost got what we'd expect as baseline sea dynamics before that exclusion zone goes. So we're not having trawling, and we know the impact from the platform is not extending beyond that. Of course, that will be site-specific and depends on the hydrodynamics of the movement of water and how that's being moved as well. But yeah, it's really interesting, complex.
Right, we've got time for one more question before lunch, and David at the back.
It's Richard, actually. Richard Thompson,
Richard.
University of Plymouth. I wanted to partly chip in to Dave's question, which is a really good one about the quantities of plastic in oil and gas related to other sources, because it is, as Sarah said, something we've been looking at. And I think, so you could compare it, let's say, to the legislation that the UK introduced to prevent microbeads in rinse-off cosmetics, and the quantities of plastic associated with oil and gas way exceeds that as an area where we've seen legislation. It also far exceeds the quantities that have been picked up by a decade of beach cleans organised by Marine Conservation Society. So it's not an inconsiderable amount. I think it's important to also recognise that we're in an era when those bad practices in other industries are hopefully going to diminish. So the relative contribution of that from oil and gas is likely to potentially become more important. Now, those other forms, the microbeads in cosmetics and the litter, aren't essential things in the marine environment. And it's those things that we need to balance. But potentially part of the issue here is the optics of what is done, that if we're agreeing to leave in the ocean a quantity of plastic that far exceeds something that we've seen DEFRA legislate against, that might not make sense to everybody. So it's about trying to weigh one thing against the other and provide some of the comparators exactly along the lines you've been saying, to try and set it into context. So it's a really good question, and we'd welcome further thoughts on that, because it then boils down to this essentiality criteria, because clearly the two examples of bad practice I've given you are about non-essential things. Oil and gas pipelines are essential, so it's how do you weigh, until they're decommissioned and they're not used. So how do you weigh those things against each other? It's really good point. Thank you.
Well, it'll be good to see discussion on this continue, and please do grab the speakers at lunch. We'll be coming back at 1:30 for the session 3, which is biodiversity effects, food webs and species interactions. So, yes, lunch will be in the main area. Thank you.
Back there.